informatics. Thank you.
Okay, hello everyone. Good afternoon, distinguished professors. My name is Zhengye Jiang from Xiamen University in China. I shall be speaking today about integrative analysis of key candidate genes and signaling pathways in cognitive decline relative to chronic periodontitis by bioinformatics. Okay, as known to all, Alzheimer's disease is a global disease with an increasing number of aging population. The economic burden of this disease on the world is also increasing. According to statistics, there are more than 50 million patients with Alzheimer's disease in the world now. And as a progressive neurodegenerative disease, it has placed in the list of diseases that threaten life expectancy in the elder. Its clinical manifesto mainly include cognitive decline. The data show that nearly 8% of all patients with AD are over 65 years old. Periodontitis constitutes a chronic inflammatory disease. During the development of periodontitis, associated complications such as alveolar bone destruction, as well as the loss of attachment of collagen fibers to periodontal ligament will occur, eventually leading to tooth loss. A large number of recent reports have shown that chronic periodontitis comprises a risk factor for neurodegenerative disease, such as cognitive decline. Among them, in the study of Castro et al., it was found that the levels of inflammatory cytokines in individuals with Alzheimer's or mild cognitive impairment were remarkably correlated with periodontitis. A large number of previous studies have confirmed that immune factors, depression, genetic factors, could be remarkably possibility correlated with the incidence and the development of AD. Despite the huge advancements in AD research, the current AD treatments can only improve and relieve patient conditions to some levels. As the treat of AD to the elderly becomes the greater and greater is the impairment for us to establish the etiology, as well as the more molecular features of AD. Therefore, we explore the molecular biomarkers by studying the correlation between chronic periodontitis and AD disease to provide evidence for early diagnosis, prevention, as well as the treatment of this disease. And how does bioinformatics analysis work? Let's briefly review. At present, high throughput sequencing techniques such as molecular diagnosis, prognosis, and estimation, as well as drug-targeted discovery, and which can be employed to access the gene expression differences, as well as the variable splicing variation, are gradually considered to the important clinical significance in disease research. The Integrated Gene Expression Database, named GEO, publicly available website supported by the National Center for Biotechnology Information, named NCBI. It covers dozens of, it covers dozens of basic experimental disease gene expression patterns and is extensively employed to explore key genes and prospective mechanism of disease, onset and development. Though the pathogenesis of chronic periodontitis has been found to be related to AD recently, its pathogenesis as well as the molecular mechanism remain unknown. Hence, we need to utilize the gene expression chip in the bulletin database and analyze its state through modern software to find new diagnostic markers and therapeutic targets. Therefore, our study conducted further research on the relationship between AD and chronic periodontitis. Next, let's look at the result of the analysis of the different genes between AD and MCI. First, let's look at AD patients. which are the heat map and the volcano map. Red indicates up-regulated genes and blue indicates down-regulated genes. Then in MCI, there's MCI heat map and the volcano map. The same red is up and the blue is down. We choose common genes between AD and chronic periodontitis gene sets. We got 305 genes. And we choose common genes between MCI and chronic periodontitis gene sets. We got 100 genes. Then we found the 3-cell annotation in the GO analysis, namely inflammatory response, extracellular space, and protein binding in and in the KEGG analysis. We also find the top 3 signal pathways with the highest enrichment, names protein galaxy in cancer, PIK3 ACT signaling pathway, and influenza A. Similarly, we also obtained 3 obvious cells from GO analysis in MCI. Note, concentrate, the same is the inflammatory response, extracellular space, and protein binding. However, in the KEGG analysis, there are 3 signal pathways different from AD, namely type 1 diabetes mellitus, inflammatory bowel disease, and cytokines and cytokine receptors crosstalk. Next, we used the string analyze search tool and Cytoscape to analyze the protein and protein international, names PPI. It is recorded by DEGS. Therefore, we obtained a PPI network containing 1,494 crosstalk based on 247 AD related integrated DEGS. According to the degree value, the first 5 hub genes extracted from the AD group were IL-6, MAPK3, VEGF-A, AKT1, and ALB. Okay, let's take a look at these genes. First, IL-6 constitutes a pleiotropic pro-inflammatory cytokine. The degradation of IL-6 is linked to chronic inflammation as well as multifactorial autoimmune disorders. The study of Gru et al. Find that IL-6 is involved in iron homeostasis and inflammation, and IL-6 was found to be elevated in the hippocampus of 5XFAD mice. A large number of studies have found that IL-6 increasing with age and is associated with loss of motor function. Meanwhile, in many mental diseases, such as obsessive compulsive disorder, acute psychosis, and a panic disorder, as well as post-traumatic stress disorder, IL-6 expression has been found to be significantly increased. Our second names MAPK3. It is critical to point out that MAPK3 constitutes a prevalent gene in most of the rich KEGG pathway in AD. Additionally, the MAPK3 gene comprised one of the hub genes uncovered by the PPI network. MAPK3, referred to as the multigram activity protein kinase 3, is the MAPK kinase family member and participates in an extensive array of biological processes, including cell proliferation, as well as angiogenesis. MAPK3 may serve as the intracellular mediators that trigger the expansion of the commular cell all-cyters complex, names COC, as well as the maturation of the all-cyters, the extracellular as well as intracellular metagenic stimuli active the MAPK3 cascade, which has pivotal functions in cellular differentiation, proliferation, and survival. A study of chronic cold cancer by Smatches et al. Showed that the expression of MAPK3 is related to poor prognosis. Third, VEGF-A is growth factor related by tumor cells It participates in tumor genesis and development. It can trigger the germination of existing vesicular cells to form a new vesicular system. It is reported that VEGF-A is abnormally regulated in various cancers and promote tumor progression. According to reports, a variety of microRNAs inhibit tumor growth. Angiogenesis as well as metastasis by suppressing the expression of its target gene VEGF-A. In addition to its widely described growth factor function, VEGF-A also acts as a neurotrophic factor, and interestingly, VEGF-A also serves a critical role in hippocampal neurogenesis. Recent studies have found that considerable evidence suggests that insufficient neuroprotection of VEGF-A can lead to neurodegenerative disease. And then AKT1 comprises a radon-sensitive protein, and its kinase activity is modulated by the Randex mailer. The result of Kendra et al. Showed that the polymorphism of the AKT1 gene seems to have an impact on mental symptoms such as schizophrenia. Dementia et al. Documented that AKT1 is a risk factor of metabolic syndrome, as well as insulin resistance. Interestingly, a large number of studies have found that abnormalities in AKT1 signal have been observed in the brain tissues and animal models of AD patients. And last, ALB. It constitutes a multifunctional platform protein found in large numbers in human blood. In addition, ALB is also involved in stabilizing colloidal osmotic pressure, removing free radicals, and protecting nerve cells, and is significantly related to systematic neutral status and inflammation. The study of Lei et al. Found that ALB concentration can be used as an independent prognostic factor for patients with endometrial cancer. And then we employed the MCODE to determine highly interconnected subnets, which are frequently protein complex as well as components of cascade as per the topological structure. We select the two most important modules from AD group respective for further analysis. Addition function enrichment assessment of the established modules demonstrated that genes in AD module were majorly abundant in the goal terms of the inflammatory response, extracellular space, heparin binding, as well as KGG cascade of PI3K active signaling pathway. Similarly, we constructed a PPI network containing 64 complete DEGs in MCI group. In the MCI group, the top 5 central genes were EGFR, IO10, IGF-1, BMP2, and LDLR. And the first EGFR function as a protein tyrosine kinase receptor not only serves important function in cell differentiations, cell growth, as well as tissue development and function, serving as an integrator at the convergence of the extracellular growth and survival signals, which are transformed into intracellular outputs. In addition, EGFR serves a vital role in the development of human cell transformation and cancer. The function of EGFR is obviously related to the series of neurometabolic disorders, such as disease, AD, as well as aging. And second, IO10 is a common gene in most of the rich KEGG pathways in MCI. And also one of the hub genes uncovered in PPI network, IO10 is a key immunomodulatory cytokine which is involved in the inflammatory response among them. The research results of Gluten et al. Propose that the rebalancing of innate immune via blocking of the IO10 anti-inflammatory response may be linked to the treatment of AD. Similarly, many studies have shown abnormal increases in IO10 signaling in the brains of AD patients. Nearby results contain and extend other studies that have reported elevated IO10 levels in serum, as well as the brain extract from individuals with AD. And third, the IGF-1 signaling pathway is ubiquitous in the aging process, so it is called the smart trophic axis. Similarly, low levels of serum IGF-1 can reduce the incidence of cognitive impairment in women after the age of 10. Then BMP2 in the study of Pending et al. Investigating apoptotic mechanism through in vivo and in vitro studies revealed that promoted bone morphogenetic protein 2, named BMP2 expression, supported by increased BMP2 and P smart 1 or 5 in hippocampal neurons. Overall, the study proves that arsenic promotes hippocampal's neural apoptosis through an upregulated BMP2 dependent attention of BDNF or TRKB pathway, inducing cognitive defects. And last, LDLR can regulate the level of peripheral and central nervous system, MCNS. So it is known as an important receptor that inhibits amyloid deposition. Yao et al. Found that polyphenol mediates regulation of LDLR. Expression may be a safe and effective treatment for AD disease, which can accelerated with the clearance rate of AD. Similarly, we employed the MCOD to determine highly interconnected subnets, which are frequently protein explorers as well as the components of cascade as per the topological structure. We select the two most important modules from MCI group respectively for further analysis. Genes in module of MCI primary were abundant in the GO terms of inflammatory response, extracellular space, chemokine activity, and the KGG pathway of proteoglycans in cancer. These are the main results of this study. Finally, let's do a brief review summary. First of all, we found through literature that chronic periodontitis is correlated with cognitive decline. Then, to confirm the correlation between chronic periodontitis and AD, we conducted a series of exploration and excavation through bioinformatics analysis. The results show that MAPK3, IO10, and OR were targeted therapy points with high potential significance. And then we found that PI3K was a potential signal pathway. This series of study and results will help improve the early diagnosis and treatment of AD. Unfortunately, because our study is an exploratory study, there are still quite few defects, namely basic experiments in vitro and in vivo. Therefore, our experimental team will explore and mine these analyzed genes and signal pathways in the future. This will provide a more reliable basis for the future diagnosis and clinic treatment of AD. Okay, the above is all the content of my speech. I'd appreciate you all for careful listening. Thank you. Thank you very much.
Okay, thank you for your talk.
Thank you, Dr. Jiang. Any questions for Dr. Jiang?
Excuse me?
Any questions for
If
Dr.
anyone has a question, please.
Any questions for Dr. Zhengye Jiang?
If there is no questions for Dr. Jiang,